Imagine if you could clone yourself, or maybe even your pet. Seems a bit weird, right? But genetic cloning is a revolutionary process. We could even use it to solve current environmental problems. But what is genetic cloning? Let's take a step back and look at genes and DNA. Deoxyribonucleic acid, or DNA, holds the information for the development and functioning of an organism. Located in the nucleus of a cell, DNA has genes that contain instructions for making proteins that perform a specific function. In genetic cloning, scientists create copies of a specific gene by isolating it from an organism's DNA. They then insert it into a carrier molecule like a bacterial plasmid. Related to genetic cloning, genetic modification or genetic engineering is the process of altering an organism's genetic structure. Let's go a bit deeper into the process of genetic cloning. A gene of interest is taken and put into a plasmid. A plasmid is a circular piece of DNA in bacteria. The new plasmid with the gene of interest is called recombinant DNA. This plasmid is put into a bacterial cell, and the new cell is called recombinant bacterium. The bacteria are cloned in a culture of cells, and the gene of interest is multiplied as well. With the recombinant DNA, unique proteins are created with the gene of interest. Scientists can conduct different research projects with the cloned recombinant bacterium. Let's look at the application of genetic cloning to environmental problems. Certain genes can be used to create bacteria that cleans up toxic waste and oil spills in the ocean. Pesticide-resistant plants can also be created to limit the use of toxic chemicals on farms and prevent runoff into bodies of water that can harm aquatic life. Genetic cloning and modification are innovative processes which scientists can use to help combat environmental problems. Through research, experiments, and application, we can create a more sustainable world.